So we talked about a bunch of opioids before, but now let's talk about some of the specific endogenous peptides that bind at the opioid receptors. There are 5 here. One of them, nociceptin, is probably a little familiar because it's the one that the nociceptin receptor is named after. So nociceptin binds to the nociceptin receptor. Now, nociceptin is actually anti-analgesic. So that means what it does is it reduces pain, it causes pain, right? Because analgesic effects means like pain reducing effects, and this is anti-analgesic. So this is what's responsible when you're feeling pain. There's only one type or one species of the nociceptin peptide, so just that's nociceptin. So now let's go through some of the other ones. We've got the enkephalins. So this is in the brain. That's what its name kind of means if you break it down. It's selective for our delta receptors, and there are 2 subtypes. Now you don't need to know the subtypes, but there are 2 subtypes. We've then got our dynorphins. So this comes from the Greek word dynamis, which means power. So dynorphins are selective for the kappa receptors, and there are 4 subtypes of dynorphins. We've then got endorphins. So these are contraction from endogenous morphine. So it's selective for the mu receptor, and there's 5 subtypes. And then we've got endomorphines. So these are also a contraction from endogenous morphine, also selective for the mu receptor. Endorphins are more strongly selective, where they have a higher affinity for the mu receptors than, say, endorphins do. And there are at least 2 types of endomorphins. We're still looking at them. So the genes and pre-peptides for endomorphins has not been identified yet. And when we're talking about our endogenous peptides in general, then you should know that they weren't even identified until after the opioid receptors were identified. And remember, those weren't identified until the 70s. So endogenous peptides weren't identified until the late 70s. And we're actually still finding, identifying new endogenous peptides. Nociceptin and endomorphines are some of the more newer, recently described and identified peptides compared to our other endogenous peptides. So we don't really completely understand their function and expression yet. Now, endorphins and enkephalins are involved in the endogenous analgesic effect. In other words, our enkephalins and endorphins help reduce pain, while our dynorphins are diverse. So dynorphins are co-expressed in cells that also express vasopressin, orexin, and oxytocin. And it actually plays a role in modulating the release of our peptides. Okay, so we've got pain relieving, modulates our other peptides, and pain relieving. And then we've got not fully sure what it does and causes pain. Okay, that's kind of a breakdown of our endogenous peptides. Now, despite some dramatic size differences in our endogenous peptides versus our natural opiates, then they are considerably more potent. So we've got morphine here compared to one of our subtypes of our peptides. So this is an enkephalin. It's one of our two subtypes, met and enkephalin. Okay, so we've got our two structures, and you can see they are so different. Our peptide is huge, right, compared to morphine. However, there's a lot of structural similarities at this one place. And that's the part of these chemical structures that are thought to actually bind to the receptors, and that's why they're able to both bind and have similar effects on that receptor. So, how do our endogenous peptides get made? Like all peptides, they start by, there's a gene that represents the peptide or the pro-peptide, and it then gets cleaved. So endorphins, enkephalins, and dynorphins are synthesized from these pre-pro-peptide genes. Like I mentioned before, our endomorphines, we haven't identified any genes or anything for them yet, so we're just going to kind of ignore that for now. Endorphins are expressed from POMC. Now, we kind of mentioned POMC before, because remember, it also gives rise to some hormones. Our adrenocorticotropic hormone we mentioned before in our neuropeptide lectures. So you can see there's some crossover in genes that express for our endorphins as well as some hormones. Okay. Now, specifically when talking about endorphins, then we can talk about beta endorphin. So beta endorphin is one of the subtypes of endorphins. And it is when it comes from POMC, right? So, because we know that hormones are usually excreted or made in our pituitary gland, then that's a great place where researchers looked to see if endorphins and some of our endogenous opioids are also made there or expressed there. And that's where we find beta endorphin. Now, we're finding it in the anterior pituitary because remember there's only a select number that are made in the posterior. Look that up if you don't remember. So in the anterior pituitary gland, then we get high expression of POMC, and from POMC we cleaved our two peptides, ACTH and beta endorphin. Now, ACTH is released in response to hypothalamic corticotropin releasing hormone, or CRH, and acts on the adrenal cortex to release glucocorticoid hormones. Okay, so from the anterior pituitary, we get the release of ACTH. Now, these hormones have to do with stress. That's what's going on here with our hypothalamic corticotropin and the release of our glucocorticoid hormones. Now, because POMC is cleaved into both ACTH and beta endorphins, then beta endorphins are also going to be released when ACTH is released. So the co-release of our beta endorphins from the pituitary provides a physiological link between stresses and pain signaling. okay? Because if one's released, the other's also released. So this is a great starting point for researchers that were like, what is this relationship? Why is there such a strong correlation between stresses and pain? And now we know it's because of the co-release of these two hormones. So, we have these endogenous opioids that are pre-existing in our body, but why do we have them? What is their function? And it turns out that in endogenous opioid signaling is inhibitory. And it's not inhibitory just because the receptors are GI coupled protein receptors, okay? There's other reasons as well. And so the inhibitory signaling of our endogenous opioids can affect the neurotransmitter release in actually a number of different ways. Now, the first way is because kind of related to and because of that GI signaling. So the first one is the postsynaptic inhibition. So we've got an opioid receptor or opioid neuron that's going to release opioids, are endogenous opioids. It's going to release it onto the postsynaptic cell. The postsynaptic cell will have our receptor, and what the receptor is going to do is it's going to activate that GI signaling down, and it's going to inhibit the depolarization of the postsynaptic cell because of GERK. Okay, so that one is because of the type of receptor. Now, the second type of inhibition that an opioid neuron can do is axonal inhibition. And this one is again related to the GI receptor. But here, then we've got a presynaptic cell, a postsynaptic cell, and then our opioid neuron. So opioid neuron is sending signals to the axon of the presynaptic cell. And what it's doing is it's inhibiting the presynaptic cell from releasing a neurotransmitter to the postsynaptic cell. And it's doing this using that GI receptor, and the CAMP signaling will then inhibit the voltage-gated calcium channels, therefore not allowing the vesicles in the presynaptic cell to bind to the cell membrane and release neurotransmitters. Now, the third type is that our endogenous opioids can actually act as autoreceptors on the presynaptic cell. So here we've got our opioid neuron that co-localizes with neurotransmitters, which means this cell releases not just opioid peptides, but also neurotransmitters. So what's going to happen is when excited, this presynaptic cell is going to release both neurotransmitters and opioids. But instead of having opioid receptors on the postsynaptic cell, then there is an opioid receptor on the presynaptic cell that the opioids that have been released will then go and bind to. This opioid receptor on the presynaptic cell will then inhibit further release of more neurotransmitters and opioids. And what this will do is it'll block activation of the postsynaptic neuron. Okay, so in this third one, then you start with some release of neurotransmitters and opioids, but then you've got it acting as an autoreceptor to turn off that release when the opioids bind to that presynaptic receptor. Okay, so 3 different ways that our endogenous opioids inhibit signaling.